Okay, so like lab book stuff for this week, when we're talking about histology tissues, you have all of your terminology up front. No. Here are the learning out objectives. This is what we make all of your test questions on, basically. There are some photos on these concepts, but this is basically that, okay? Down here a little bit more is just what we kind of went over today. It's the readable part of all the information you need to know. It's the handbook. We use the OpenStax, and if you like kind of click into it, I think it's right here. So if you click into all these links, it'll take you exactly where you need to go in the OpenStax textbook. Hold on. I've got to like click a million things, I think. All right, hold on. Thank you. Okay, there it is, right? Here's the OpenStax textbook. So that if you don't remember exactly how things went from lecture, it's okay. It's going to be okay. Okay. Where did it go? Where did it take it? There it is. Okay. That's not the right one. We'll go back. There it is. Okay. All right, so it'll take you to where you need to go. Also, I have some videos uploaded. And, I'll upload the ones I did today. So here they are, the basics, couple of questions. You might need to remember a few things from biology. You'll need to know about like what an endoplasmic reticulum does, what a nucleus does, what's a cell membrane do, right? So go back and definitely review those. There's also videos on our site for that if you need that. So these are important for these are really important. So I would, if you can, print these out, and I would do color if you have the option. And here at the school, like the print center that we talked about, the DPC can definitely do that. I think it's like 30 cents for like color. Maybe I'm wrong. I would check their website. Because what you're gonna need to do is was it super easy to remember all of if I gave you a test right now on those tissues, would you be like, got it, no problem? Or would you be like, there's so many squiggly purple and pink lines? You would probably say the latter. Okay, so what we have to do is you have to kind of train your brain on the minute distinctions. And so this is an example that I used like a million years ago in some of my research. And this is a dichotomous key. So I don't have a dichotomous key for you. You're going to build one. Let me explain what a dichotomous key does. It tells you here is a difference between some of these very similar items, species, in your case, cell types. What are the differences that I'm looking for? And then you follow the clues until it takes you to say, this is this species, or this is this cell type. I'm bringing this one up because let's talk about mosquitoes, for example. A dichotomous key for mosquitoes. So malaria is a pretty nasty parasite-driven disease, okay? But not all mosquitoes will give you malaria. So if you go to a place with a certain type of mosquito, you're going to want to maybe take chloroquine before. And it makes you very, very sick, I can tell you. I've done that. It makes you very sick, and it's horrible, and the headaches, and the stomach, and everything. So you kind of want to avoid it if you can, because it's not super nice to your filtering organs either. But if you're going somewhere that doesn't have those mosquitoes, maybe you don't need to worry about malaria. Does that make sense? So here's what we could do. We go out and we just trap a couple of mosquitoes in a little net. And then we put them in a freezer, and they get really cold, and then they just kind of die. And you can look at them under a microscope, and you can check a fine detail to know whether or not you're in danger. Okay, and that's what the dichotomous key can do. Here's a mosquito head, and it has some antenna, and here's the stinger. Oh, there it is. So here's the head, here's a couple antennas, here's the stinger. We call it the proboscis. But then it has these little extras right here. Those are called palps. If you are going to get bitten in an area and get malaria, your mosquitoes are going to have some long palps. They just, for some reason, have a body type that can carry the parasite, but then these ones don't, for example, in our scenario. You might get West Nile virus, you might get a different encephalitis, but you're not going to get malaria. Does that make sense? And then you can kind of further go down the line. You can say, well, am I really going to get encephalitis, or am I okay for that risk? So you can kind of go down, and there are other features that you can look for. Okay? You can say, oh, well, this has this kind of like body type, or this one has little hinges on its abdomen, or this one has a spot on its wing or an extra vein on its wing. It's a key. You start with similar things, like all of the cells that you're going to look at, for example, will have nuclei. We hope. But they're going to look different. Some of them are going to look like little eyeballs, you know, maybe. Or maybe they'll be arranged, like maybe they'll be very flat, right? Or maybe some of the, so there's going to be all these little things. Maybe there's going to be large spaces because it's in a single layer. Or maybe it's going to have to be like, relatively thick because like this is a very thick stratified layer, right? Because on the outside of your skin. So you're going to be looking for those kind of different things to differentiate it. And so you would take these and you can build a dichotomous key from that. Okay? Here's more. Oh, these look pretty different from the other ones we were just looking at, right? So one of your things would be, does this cell type have large white circles containing lipid droplets? Yes or no? If yes, then you have adipose tissue. If no, then you go to the next one. You say, okay, it doesn't have that. Okay, well, does it have squished nuclei in a long, like kind of pink wavy field, or does it have open nuclei and lots of crisscrossing fibers with like big white open fields? And you're like looking down, you're like, oh, there is no open field in here, and all those nuclei are squished in a pink field. There, okay, I must have a ligament or dense connective tissue. No. You can screenshot some of these and put these together. So there's those. So that's kind of like what you do on your own. Is this going to take a lot of hours? Oh yeah, oh my gosh, this is seriously like a 4 to 5 hour situation. And like, it's just, like, this is probably one of the hardest things, I think. So here's some extra key items that you can put in there. It helps you get used to where they are in the body. Here's the instructions a little bit more based on kind of what I talked to you about. Look up Dichotomous Key. I liked this one from the CDC. It goes through all kinds of different things for mosquitoes, so you do a similar thing for tissue. How do they look? Is there a color difference? Are their heads different? Is the cell, what do the nuclei look like? Okay. There is, oh my gosh, an alternative assignment. Did you guys just like stop sweating when you saw that? I mean, it kind of is. I mean, you can find some of these different tissues, maybe make a quiz for yourself, but show that you have some of these out. Do like maybe simple drawings. Put a lot of effort into it because I know how long it takes to put a dichotomous key together. That's like a 5-hour situation. So like I want to do like a 5-hour situation on this because you need to know this if you're going into healthcare. There is no she's being mean. This is like basic requirements nationwide. And it's fundamental for every single chapter we'll do. So it feels hard because it is hard, but that's okay. Because you're tough. You're going into medicine and you're ready to do the work. And if it feels overwhelming, then you're feeling the right emotions because it did that to me too. It did it to everybody. And if you have a hard time, still put in the effort and then come in and we talk about it and I help sort you out a little bit and get you on the right track. Does that make sense? You're going to feel overwhelmed. It's going to feel hard. You're going to say this is a long time. You probably will want to cry, and that's okay. I've been there too. Everyone is. And then you come in and talk to me, and then I'll show you how close you actually are just to being in great shape for it, okay? But if you bring me work that doesn't show that you put in the effort, it's gonna show when I kind of start talking to you about it. Does that make sense? Okay, you know, don't stress too much, but yeah, it's stressful, so it's gonna be there. All right, let's talk about the rest of the activities. Okay, so we talked about making the dichotomous key. So for lab 2, histology, we're making the dichotomous key, or you can do the alternative, which I think is actually harder because you have to come up with it by yourself. The next one is going to be cell size, predicting cell size. So if you remember, we had in Bio 1, one of the first few labs, we did the thing where we found out what the total magnification was in micrometers. And then you say, like, okay, well, this cell could kind of fit across it about 10 times, about 2 times, about however many times. And then we divide those numbers together. So this is essentially it. So remember this equation for estimating cell size, or you can also super Google it. It's okay, or we can talk about it. But I'd like for you to try to remember it, because it's important for you to have that recall process, okay? So we'll take a couple of pictures, or a couple pictures are provided for you guys here in the lab. For all of you folks, you get to just look in a microscope and you can take a photo. So we can set you up with a different tissue type, and then after I get it focused, you can actually just go around and take a photo on your phone camera. I'll show you how to do it. It's really nice that it does that for you. And you can draw it, or you can just provide the printout and label all the structures we talked about, like the nuclei. If you got some collagen fibers in there, maybe an open field, right? So talk about the matrix a little bit and what you're seeing. Estimate the cell size. And then talk about it. Explain why or not your data supports your hypothesis. Like, I believe this is accurate because it has these features. Here's my dichotomous key. It has all of these features in line. And then more importantly, it does not share these, like, it does not look the same as these other things that are kind of similar, right? Like, also weed out what's in the neighborhood. Okay, sound good? It sounds tough. It's okay, I'm here. And we're gonna do it together. And it's gonna feel better after you get your feet wet, okay? All right. Any good questions?